are involved in this outbreak, and I tried to spend this half an hour outlining the molecular test and their interpretation. Today, I'll cover the origin of this virus and the molecular structure that is useful for explaining you why the molecular test has been developed. I outlined the critical role of laboratory medicine in this field, some points of the IACC interim guidelines on this type of test, some points outlined for the molecular testing, the variant diagnosis. You know that there are a lot of variants now in different countries, so this is important also to detect which variant is prevalent in a country, and finally some conclusions. This is the story that started at the end of 2019. SARS is start from some bats, and this is the structure of the virus, is the classical coronavirus that I'll describe later, and the human being is the intermediate host. This is really an important point that also I would like to already suggest that this is the third coronavirus for some outbreak. It started in 2019, officially the 17th November 2019, and this is the third coronavirus in the last 20 years. Before, there was the severe acute respiratory syndrome, SARS, in 2002 and 2003, and the Middle East respiratory syndrome, the MERS, in 2012. On the right of the slides, you may see the dimension of the different viruses and how they are important in the history. This means that virus was always present in the human being, but we hope that more and more will be able to fight with success. This is the SARS-CoV-2 is an enveloped virus with a positive sense, is a single-strand RNA genome containing 4 main structural proteins that are known as a spike, the S, which contains the receptor binding domain, known as RBD, the envelope that is named E, the membrane M, and the nucleocapsid is N, along with other additional genes such as the ORF1AB, the ORF3A, the ORF6, the ORF7A and B, the ORF8, and the ORF10, which encode accessory proteins, including the RNA-dependent RNA polymerase. In the right part, here you see the dimension, the genomic size, and the protein size of this different part of the virus. This is to move more in the structure. This is the schematic representation of the Genome Organization. The virus encodes around 28 different ops that perform functions related to viral genome replication, the virion formation, the virus entry, and immune evasion. You can see here the structure. There is the operating frame 1A with some region for the protease production. There is ORF1B with the helicase and the RNA-dependent RNA polymerase. There is the virus binding and entry. This is the most important part of the virus, the S. And in this part, there is the other operating frame. There is the envelope, there is the membrane, and there is a nucleocapsid. This is the structure of this genes, and you see that is sufficiently complex, but it's clear how to approach the diagnosis of this virus. This is very important, these slides, because it's the public health impact and the laboratory medical consequences in COVID-19. The period is from the beginning, January 20, up to the half of the September 20, and you see the different localization. You see that it started at the beginning, for sure. This was the Western Pacific country. This was Europe. This is the Americas. And you see that this is the first wave. This is the second wave, and then later on we know that we had already the third wave. This is for sure is a global dissemination of this virus. The WHO declared a pandemic in March 2020. And there was for sure a lot of problems for the life of all the people in the world. What are the laboratory medicine consequences? For sure, we have to start with the genome sequencing. A clinical assessment is really very important. In this part, what is really mandatory is the quality of the assessment. And in this type of approach changed also the approach in all the world with the emergency use authorization that has been planned by both the FDA in the United States and also in the economic community, because we need to approve very fast all the tests. All this type of design, development, validation are focused in molecular testing. That is today discussion in this presentation, but also in serological tests that we don't discuss today. And you see that in the molecular testing, we have to design the primer probes of the assay. We have to develop the internal controls. We have to optimize, possible to develop automation. There is the industrialization of the kits. There is the clinical validation, the launch of laboratory test, and the launch of point-of-care test. You see that this is the first European approval was in March 2020, and for the serology, the 1st of April. You see that in a very few time, we already approved several tests. The test is to have the availability. We need in the laboratory to improve the implementation, to implement the availability of this test. We need mass production, extensive transport, broad implementation, the aggressive search and destroy, and the storage for future use. You see that is really very complex for laboratory. Then I would like to outline the critical role of laboratory medicine in COVID. You see that after the infection, the first step is for sure the diagnosis. The infection is in charge of the over disease, and in this case we may recover, or there are a lot of death depending on this virus. For sure, is important different activity. For diagnosis, the gold standard is the molecular test, mainly the RT-PCR. Is also very important for the epidemiological surveillance, both molecular and antibodies test. During the disease, there is the clinical approach of the laboratory with the staging, prognostication, and therapeutic monitoring with several biochemical tests. And then there is also what happens after the infection with the antibodies surveillance. This is really important to know the kinetics of virological markers to understand when is the best period to detect this virus. You see that this is the contact on the left with the virus. It is really very important. This is the symptoms onset. And you see that depending on the different sample that we collect, nasopharyngeal or bronchoalveolar lavage, or stools, or other type of samples, you see that we may have more or less the peak of the viral load at the beginning of the first day of the onset. You see that then they decrease slowly. I look at the blue light curve because it's nasopharyngeal that is more popular. You see that increase during the pre-symptomatic period, reach the peak, and then decrease during the days. Later on will appear the antibodies. You see that this we may say that is detectable depending on the sensitivity of the method as a day one of the symptoms, but the peaks is within the first week of the symptom, after the symptoms, and then the decline after 2 or 3 weeks after the starting of the symptoms. To know what happens, you see that this is only a short period, the beginning of 2020, in which is the number of publication of the molecular testing and the number of tests that's been performed. You see that there was an unbelievable growth of the test that is really very important for detecting all the affected people. Some definition by the WHO, what we may define a confirmed case. The confirmed case is a person with laboratory confirmation of COVID infection, irrespective of clinical signs and symptoms. This means that if you are positive, you are a confirmed case. Usually the routine confirmation should be based on detection of the unique sequences of the virus RNA by NAT, nucleic acid amplification test, and the most popular is for sure the real-time reverse transcription polymerase chain reaction. Sometimes we may confirm this data also by sequencing. Finally, the target of this test are the regions of the viral genes that include the N, E, S, RN, RD, RP genes. Some practical indications to minimize the risk of diagnostic errors in identifying severe acute respiratory syndrome infection. For sure, it's very important that the clinical and epidemiological evidence, the probability of the exposure, the signs, the symptoms, the negative diagnostic test, especially for other respiratory illness. This is really clinical epidemiological evidence is important. It's important also to chest computed tomography. This is really one of the tests that you may add to the molecular to confirm. Is important to recollect and test upper respiratory specimens in patients with negative RT-PCR. When this is negative, we have to recollect and test other samples. And when there is for sure high suspicion and probability of SARS-2 infection. Is important to provide a clear instruction how to collect the samples. We need the compliance with the assay procedure. We need to have in laboratory quality assurance. We have to validate extensively in advance the test, the clinical use of the test. And also, eventually, we have to refine molecular tests, like, for example, when there is some new viral strain. This is our other recommendation, always by the IACC, and the clinical indication for molecular testing for the SARS-CoV-2 infection. The following indication should be regarded as supported by current evidence of the clinical value. First, is important to diagnose the viral infection in the acute phase of symptomatic illness. This means as soon as possible to avoid the dissemination of the infection. It is important to assist in clinical assessment of asymptomatic, pre-symptomatic, or mildly symptomatic patient with known exposure to positive COVID-19 cases. This is important because there are about 2/3 of the patients that are asymptomatic. The molecular tests are really very important. It's important to assist in screening of asymptomatic, pre-symptomatic, or mildly symptomatic individuals in various contexts, like, for example, prior to scheduled surgery or delivery, enter the hospital, for travel, for hospital discharge, to return to work or school, and to manage a small outbreak. Some comments regarding the importance of the assay methodology. There is no doubt that NATs are currently the gold standard for diagnosis suspected of the infection. RT-PCR is the most common type of NAT, and are applied in authorized molecular assay and is used by both the Center for Disease Control, the CDC, in the United States, that developed an assay, and also by WHO that endorsed an assay. There are other approaches, for sure, that are less popular, like isothermal nucleic acid amplification, such as reverse transcription loop-mediated isothermal amplification, the LAMP, transcription-mediated amplification, and also the CRISPR, that is cluster regularly interspaced the short palindromic repeats, and that are the other popular. We'll see in the future slides that there are a lot of different tests that we may utilize for this type of approach. Another recommendation is the importance of the viral target selection. There is currently insufficient evidence to suggest a definitive advantage of selecting an assay based on a specific SARS-CoV-2 gene target. Different kits may have different targets that are also different from one part of the world to the other, in some parts at least. But what is important to say that the minimum of the 2 gene targets are needed to be tested to minimize the risk of false negative. It's important to avoid false negative. 3 targets are also better. Also, is very important the specimen type selection. We need that acceptable specimen type for molecular testing should follow manufacturer recommendations. It's important to follow how they say, how they test, how they validate it. At minimum, we need an upper respiratory tract specimen for the first diagnosis, is the most popular. There is some evidence that we may add some time. We may utilize also saliva as a sample. Now is more and more popular. The self-collection kits are not really recommended unless there is appropriate instruction or patient education, and all the results should be interpreted with caution. And also in some parts of the world, not in Italy, for example, they utilize also pooling specimens, like also, for example, in China, but should be utilized in low prevalence area, at least with less than 5% of prevalence, but is preferable with less than 1%, or where there is a very low resource setting after appropriate validation. Some points that are really important in molecular testing, outlining the pre-analytical vulnerabilities. This is really very important. In the pre-analytical phase, one point is the lack of identification or misidentification of the sample. When you have 1,000 and 1,000 tests, this is very important to have some computer-driven organization for detecting all the samples. We may have some inadequate procedures for specimen collection, handling, and transport and storage. The swab are really very important to collect. There is sometimes collection of inappropriate or inadequate material for quality and also for the volume. There is sometimes the possible presence of interfering substances, and also we may have some manual errors in pipetting or preparing the samples. Specific pre-analytical problem, not general, are the sample contamination or testing in patients receiving antiretroviral therapy that may interfere. Also is very delicate the analytical phase. The testing should be carried out outside the diagnostic window. If you do the test too far, this could be a problem. We may have active viral recombination that may interfere in analytic test. We may use non-adequately validated assay. You know that there was a rush in producing kits. There are hundreds of different kits, and not all they are so valid in doing the test. There is, if possible, the instrument malfunctioning. There is insufficient or inadequate material. There is no specific PCR annealing if the probes are not very specific. And also there's a problem of misinterpretation of the expression profile. This means that we have a lot of possible problems in pre-analytical or analytical phase. This is the mandatory activity of the laboratory. How to detect the RNA? The standard is the nasopharyngeal swab. We have to target at least two of different regions. And what is important is to have, we need 3 or 4 hours to perform the test. This means that we should have the most fast organization to have the final result in a short time. The performance of the kit are usually satisfactory. There are, you see, this is a publication regarding the sensitivity and specificity of the molecular test. 2 comments, both are really very good. There is variation among different kits. But what is really important that the limit of detection of the copies could be different in different kits. This is important when you compare data, because utilizing different kits, you may have some kits that are positive because are more sensitive, and others are negative because are less sensitive. This is important. If you repeat the test, we must repeat with the same kit. What is really important in laboratory medicine? We need to simplify as much as possible the procedures. There are also for some parts point of care testing. This is really very important. We may simplify the sampling like utilizing saliva. We may also pull some sample when you have to screen a very large population. There are good results regarding saliva. Not all are agreed that is better in comparison with the nasopharyngeal swab. Roughly speaking, they are more or less comparative. In our hands, is a little less sensitive, but we may utilize particularly in pediatric age. It's really important to outline that different sample means different type of sampling. We may have a different detection of viral load. You see that nasopharyngeal or saliva, they have the highest viral load. Sputum, the bronchial lavage, the stool, the serum are different. You may also, they are different, the peak in the different sample. And also you see there is the detection rate that is different. This means it's really very important to have in mind all this data when you analyze a different sample. This is, we have no time for sure to touch all these points, but you see that this is all the nucleic acid-based COVID-19 diagnostic test. There are all the group of non-isothermal amplification with a different type of test from real-time to endpoint PCR with the digital PCR. But also there is the isothermal amplification in which there is the classical lamp, and you see up to at the end, you may also sequence with Sanger, with NGS, our sample. This means we have really a huge amount of possibility in doing tests, and for sure is important that the laboratory performing this test are very skilled in several of these type of testing, and they may confirm or they may study better the sample if there is some doubt in the results. Okay, what is the significance of prolonged PCR positivity following the recovery? There is some data that suggests that around 15% of the COVID-19 patients retested positive after discharge during 14 days of strict quarantine. This means that why they may be able. One is that it's possible that the second PCR during the quarantine is negative for some, it's like false negative. This means is only the kill of the positivity. There is a strong data suggesting that the positive signal of the viral RNA might be from the dead viruses of viral gene fragments without active viral replication. There are in vitro studies that outline that the replication of the virus stops after a couple of weeks, more or less. There are some delay in virus clearance in some patients, depending on some therapy like glucocorticoid. There are comorbidities in the older patients. Few words in the last 3 minutes outlining the COVID-19 variants, which is the importance. We may say that small changes that may increase the fitness of the viruses, and this may escape from the immunity. Changing this structure, they may skip really our control. There is less common than other RNA viruses. This is important. And there will be a big question for SARS-CoV-2 going forward. What happens in the future? There is still a question mark. Another point is to outline that the spike protein is the most common and important target for this variation. Variation may happen in all the different parts of the virus, but the spike is necessary for virus infection at entry. It is the main target for antibody immunity and also for the vaccines. This means this is the major part of that we have to study to understand what happens in a different country. What is important is that we know that other viral protein may alter host interaction viral processes, but this take a longer time to develop. Some data regarding, we don't enter in all the different variation, but you see that from the Wuhan market, that is the supposed start of this pandemic, you see all the different variations that appear with different clade of the virus, up to the American, the Indian, the UK, the Brazilian, the South African variants. You see that during the time the virus change is doing that to survive, to skip the control. You see this is the models of the protein so far. Not all the proteins has been defined, but these models, informatics model. And you see that all the different change may change the aspect, and then for sure to control by the vaccine is a little more difficult. This is the distribution of the different lineage in the world. You may see that is really important to see that some color are more present in another they are not present in other parts. This means that the lineage distribution is very different from one part of to another part of the world. This means that to try to control this type of pathology, we may solve the problem all the world. Otherwise, other variants may reach other parts of the world. Finally, which is the utility of the sequencing of the various COVID-19 strain. This is really important for the pathogen discovery with the different methodology, identifying novel pathogen or the inside in the mutation, the spike protein, and so on and so forth. Is really very important for the clinical diagnosis, because you may define which type of strain is prevalent in a specific area. Is very important for the outbreak surveillance, which is the prevalence of a specific variation in a country. You know that we in Italy, for example, we moved from the classical Chinese to the European, with probably Spain origin, then to UK, and now with the Indian. This means it's really important to evaluate all this data, not only for the surveillance, but also for the diagnosis, because sometimes you have to know that the variation may impact on the efficacy of the molecular test. This is the final slide, is the complementary role of this, all the diagnostic tools in the different phase of the pandemic. You see that in the pandemic phase, for sure, we need the scale up of the testing. We need the commercial platform with the high throughput for doing a lot of tests. We have to develop alternative reagents and protocols for improving the test during the time. We have to have stronger extraction protocols to avoid any problems in doing the test. We have to collect all the respiratory samples. We have probably to look how to develop also at home cell collection for controlling the population, and also the antibody test to measure the seroprevalence. Before the pandemic and after is really very important. At the beginning, the novel sequencing and novel pathology discovery to understand which is the problem. And after, for sure, we have to be able to larger scale population while screening to control all the infections. Okay, I am at the end of my time. I would like to thank you very much for your attention. I hope to clarify some points. For sure, the laboratory medicine was one of the major players in this outbreak. And in parallel with the clinical activity to solve the clinical problems, the laboratory medicine is was really mandatory, is still mandatory to make a diagnosis, to follow all the different spread of this infection. And I'm very happy to be here again to present this data in your Congress.
Okay, thank you very much for the very good presentation from Prof. Revar. I think we are going to continue the next speaker, and then we are going to have for the participant for questions. Now the second speaker is Dr. Miswar Fattah, MS.i. His education from SMAC Depkes Makassar, and then chemistry from Hasanuddin University 2002, and also he got a Master's of Science in Clinical Chemistry Biomedicine from Hasanuddin University in 2006, and also he got a doctorate degree of medicine from the same university in 2012. And present educational activities, quite a lot, specialty and research laboratory manager, Prodia Clinical Laboratories since 2018 till now, and Patel Key as a vice president in 2017 till now, and member of Collegium Patel Key 2015 and now, till now. And in IACC as a member of reference interval and decision limit, Indonesian Association for Clinical Chemistry 2013 till now, and president of Asian Association Laboratory Scientists AACLS 2018 to 2020, member of board of directors of Asian Association of Medical Laboratory Science, AAMLS, 2017 till now. And the last corresponding member, Scientific Committee Asia Pacific Federation for Clinical Chemistry, APFCB 2010 till now. Now, time is yours, Dr. Miswar, for your presentation for 30 minutes, and time is yours.
Thank you very much, Dr. Demak, for your kind introductions. I'm glad today because we discussed with Dr. Demak and Prof. Maurizio Ferarri. I remember 5 or 6 years ago, we discussed with Dr. Demak in Darmais Hospital with Prof. Ferarri to make a course related to molecular diagnostics. Thank you, Dr. Demak. I will start, I will share my slides. Okay, I hope my slide, you can see my slide. Okay, I will discuss about the pitfalls of COVID-19 molecular testing and interpretations. Actually, some slide is similar with Prof. Maurizio Ferarri, but we can discuss more technical about what the pitfall of COVID-19 molecular testing and interpretation. This is condition in Indonesia. Now we are 18 country, the number case above 2 million case. And today we have higher case. So in 5 days, the case increased very drastically. And now we pass from, we can say second wave or not, I don't know, but this is a higher case compared in January, only 14,000 case, and in the yesterday we have 21,000 positive case. So this is a number of dashboard of Indonesians. Indonesian molecular testing is government divided by 2, PCR and PCR. The number of tests is yesterday is 850,000. So the volume of the capability test in Indonesia is very high related to molecular testing. This is increases of molecular testing capacity in Indonesia. We changed from the April, even in the beginning of the pandemic, we start only 32 laboratory can doing molecular testing, especially for COVID-19, SARS-CoV-2. And only 3,000 samples per day national Indonesia can cover as a capacity, total capacity as national. And we have a lot of transition molecular testing, so very huge difference, very drastic change in 13 months. Now we have left from 32 to 830 laboratory can running molecular testing related to SARS-CoV-2. And from 3,000 to 850,000 capacity of the number of tests per day, the capability of total laboratory. And if you look at based on this distribution around Indonesia, already registered by governments, and we have a little of to know how many instruments they have per laboratory, mostly. Around 45% they have only one instrument. Some they have 2 instruments, some laboratory they have 3 instruments in their lab to running molecular testing. So because the drastic change of the molecular testing in Indonesia, so we need to prepare how to make a good laboratory practice related to molecular testing, and we will discuss today about that. And this is racing to develop COVID-19 tests, so we can know the impact of molecular testing from HA tissue, electron microscope, the viral cases, and NGS. After NGS already sequenced in the Wuhan, and now the sequence already after they published by Professor Yong Zhen Zhang, they published the sequence and they give name of the Wuhan first name, it's complete genome. And after that, we can doing real-time PCR. And after that, we can protein isolation, specific antibody, we can do ELISA and rapid lateral flow and chemiluminescence. This is a very important next generation sequencing and molecular testing as a standard tools for diagnosis SARS-CoV-19. And based on next generation sequencing, so we can mapping the whole genome viral of SARS-CoV-2. So we know the structure of a gene, and we can choose which target they have very specific for SARS-CoV-2. So we can define that, to develop testing related to SARS-CoV-2 with high sensitivity and specificity, depend of what technology we have and depend what specific target we have here, and very specific. And this is different type analyte we can available related to COVID-19, related to RNA. We will discuss today related to more specific for molecular. A lot of target available in the world now, like for RNA, we target RDRP, ORF1EB gene, N gene, E gene, or S gene. We can utilize a lot of different kind of technology, from real-time PCR, LAMP, or NGS. Prof. Ferarri already discussed about it too. And we can continue with what the etiological diagnosis SARS-CoV-2 infections. The two points very important. First, we need to collection an upper respiratory specimen, like a nasopharyngeal or oropharyngeal swab. And we need to run with real-time reverse transcription polymerase chain reactions. So with these two kind, we can etiologically for diagnosis of SARS-CoV-2 infection. And IACC, because based on the we discussed before, a lot of target of genes, and IACC already have a recommendation about the gene target selection, what target we can do as a target. Based on evidence, there is currently insufficient evidence to get definitive advantage of selecting and assay based on a specific SARS-CoV gene target, like for E and RDRPE or ORF1EB gene. So the IACC recommendations assay for molecular diagnostics should employ a minimum of 2 gene targets. So this minimum, the main target is to minimize the risk of false negative. This is very important. And if we look at Indonesia situations, they have variation target gene to in Indonesia. We have already surveyed from around more than 100 laboratory in Indonesia. Mostly they use a target as N protein, as N gene as a target, and RDRP and E gene, so the combination of the because a lot of laboratory in Indonesia using we call Indonesia CTM, because distributed gene expert around Indonesia is a lot of too. So this lab kept running, and we know this CTM gene expert, the target is gene A, gene I. So this is population increase too in Indonesia. This is a variation of target gene in the first time. It's why after we know a lot of differentiate target around the world. So the IACC and WHO to have a guideline to how to choose the targets, the minimum 2 targets to utilize to reduce to minimize the false negative. And what technology available related to real-time PCR? We have a lot of technology. Prof. Ferarri already discussed too. This real-time PCR or isothermal, we can use NASBA, LAM, Sherlock, or detect. So LAM and Sherlock has now increased the number of tests, real-time PCR, and some endpoint PCR to increase in Indonesia. The utilization of this technology increase in Indonesia. So if we look at the general step Indonesia as not only for SARS-CoV-2, we start from DNA extraction and PCR, real-time PCR, and gel extrapolations and sequencing. If we're using real-time PCR, mostly for diagnostic tools, we only doing DNA extraction and real-time PCR. For gel electrophoresis and sequencing, we utilize only for confirmatory or for research based, not for routine. But now with NGS, some NGS based can be as a routine tools, not only to detect the variant, but only because they have sensitivity similar with the real-time PCR. So some laboratory utilize NGS as a routine testing. And if we want to develop molecular testing, we need to utilize guidelines. This some guideline available from Clinical Laboratory Standard Institute. We use number M19A to establish molecular testing in clinical laboratory environments. This guideline is very important tools to how we establish molecular tests in our lab. And a lot of guidelines available in CLSI, especially for infectious disease, because this is SARS-CoV-2, we can utilize MM03 as a guideline in the flow of this test. And before we start molecular testing in our lab, we need to prepare our knowledge in our team because a lot of steps actually in molecular laboratory. So this is step we need to prepare before we run it. How we select the method, because a lot of methods available in the world now, especially related to SARS-CoV, and how to validate or verify this test in our lab. If they have a home room-based testing or we develop by ourselves, LDT, we need to check electronic PCR or primer probe checks to want to assess the specificity to SARS-CoV-2, and how to doing samplings, because the sampling is very important. Prepare the sampling, extract the sample, how to running PCR. This is if we need to RFD, but for SARS-CoV, we don't need this. And a lot of steps, maintain quality, how to interpret real-time PCR code, because this is very important. And bioinformatic compare gene bank, because mostly laboratory, if they utilize homebrew or he designs this test, they need to monitor impact of variants to his test. Yes, also we need to prepare this. And how to report the results, because we need to follow the guideline. Some guideline for WHO, some guideline for professional body in this country. So we need to prepare based on follow of the guideline. And we need to prepare how to summarize daily, monthly, or weekly report, so we can monitor this result day by day, and how to communicate the result to a patient, how to communicate, especially for clinicians, and how to communicate limitation of our test is very important. How we need to important tools to communicate to other laboratory. There is two types of real-time PCR for SARS-CoV-2. One-step real-time PCR and two-step real-time PCR. But mostly because the number of tests is very high, and we need to only measure a single time, not measure for multi-tube in one running. So we can utilize mostly in SARS-CoV-2 detection, we use a one-step real-time PCR. And what the critical factor in molecular testing, because this is very, very important if the lab just start to running molecular testing, we need to know about the RNA degradation related to molecular, because this is potential to false negative, and PCR inhibitor, because some, if we monitor the result, a lot of invalid because PCR inhibitor, internal control not become more positive because they have a lot of inhibitor. So this is potential for false negative and related to low quality of the reagents, because very rush we need to found, especially in the beginning of the pandemic, it's not easy to get the reagents. And so because very limited, so we chose based on what kit available in the for our lab. So it's very important about this too. So the low concentration of RNA, this can become a highly variability of result, and pipetting error, contamination, quality PCR tube of low quality of thermocycler is a critical factor for molecular testing. And to design laboratory is very important too. They have to type how to design the laboratory, 4 rooms, 3 rooms, or 2 rooms, depend of the how closed system or instruments. Is better is if we have 4 rooms, especially if our lab is they are not only for SARS-CoV or for another test in that lab. So we need to very clear separate to avoid the contaminations, especially if we have post-PCR. But for real-time PCRs, only not need to continue with other tests. We separate, can separate by 3 rooms for reagent preparation, nucleic acid expression, and amplification and post-amplification we can combine. If we have very automatically, we can separate only by 2 rooms for closed system, for preparation, especially for reagent and nucleic acid inspection and amplification and amplification. And some laboratory, the nucleic acid amplification is one setup instrument, so we can separate only by 2 based on this. And we need to know the potential source contaminations in PCR and how to avoid. We can avoid with laboratory designs, laboratory practice with SOP, and routinely to maintain our lab with chemical or enzymatic control. And this is a workflow of the lab we need to prepare in related to how to develop a lab, because very important to avoid the contaminations. Because if we have 4 rooms and we know post amplification, they have amplicon there, and if we not separate the room and not unidirectional, it's become highly potential cross contamination is developed. Especially if we have only low volume test and we have a lot of samples, this is very highly to potential contamination in our lab, because a lot of sample will be pending in our lab. And the caution PCR contamination is very important, so we need to prepare a lot of how to maintain, especially to maintain the airflow in our lab. And because this is mostly if we running with manually, it's very different with another test. We need to pipetting with very low volume. So this is a pitfall in the lab, because a lot of technicians not familiar with this. So we need to train this very best if we're running to very manually. The same. And how to choose laboratory is very, choose test method is very important, because now in the find database available more than 630 reagent available, and a lot of reagent is not very well known, and related to molecular they have increased too. So the first step, if we want to running molecular testing, we need to make a comparisons, because this is based on available kit in Indonesia. In the first beginning, we need to know the country of the producers, gene target, LOD, CT cutoff, PCR platform, match current facility, open or closed system. This is we need to know based on our laboratory. And 2 types of instruments: the closed system and open system. Closed system is for extraction, amplification, is dedicated, and sometimes they very automatically to interpretation. Open system, we can mix and match for some reagent available. Examples for closed system, this is some example closed system, like from able to separate gene experts utilize three targets: Rose, Solgen, and Ferarri's laboratory. And for open mix, some reagents available too. We need to choose primer and probe. We can make master mix from tech polymerase, what type or reverse transcriptase we can utilize, PCR or control, so we can combine in one lab. So this need to highly validate it in our lab. This is examples, some open system, like for maybe in Indonesia, like for Bamerius, BGIs, Daan, and for Perkin Elmer, but they have closed system too. Primer design, SD by sensor, tip module, SD by sensor. This is a thermo PCR. And for real-time PCR, we need to very understand about the what chemistry they utilize, mostly in using hydrolyze or Pacman probe. And we need to know the output of the PCR testing. Yeah. Output of PCR testing is, this is input, the reagents, the output actually as a curve. So we need to know how to read the curve. Yes. If you look at the cycle PCR, real-time PCR curve, this is number of cycles, because PCR based on number of cycles, and fluorescence intensity. If the DNA is higher, so they can detect by instrument in the early cycle. So it means more DNA or RNA, or they have a very late, so this less DNA or less RNA. And we need to know this is how the instruments they change, because we from raw data and make mathematical simulation to make this curve more smooth. And we need to know, we need to define or producent reagents define the thresholds. If they have a closed system, if the open system, we need to define by ourselves to validate it, this. And we need to know about very important because this is if we have more than 3 type instruments, if we have more than 2 type instruments, different instruments, they can be different, calculate baseline and threshold and determine CT value. It's why each instrument use different algorithm for CT calculation. It is possible to get different CT value from same samples. Same sample, one sample we run in 2 different methods, then we can become a different CT value, because one factor is different how they calculate the baseline and thresholds. And the possibles get a CT value from same sample, same instruments, but they have a different analysis algorithm. Although CT value can vary between positive samples, so the character simplification proof PCR negative control. And visual inspection of the amplification proof is very important. It's why if the instrument is, we can look at the calibration proof is very important, because some instruments like a black box, we can look at the proof as a laboratory is need to look at the amplification proof. And because each reagents or methods, they have different cutoff. Some laboratory they use 35 cutoff. Some method they use 37 cutoff or 40 cutoff. We have a survey from in Indonesia. Mostly cutoff is less than 40 as a cutoff, around 55%, but a lot of variability. Some laboratory they have 34 cutoff, 35, 36. This is very high too, 70%. And 38%, they have some different cutoff. And what factor affecting CT value? A lot of factor, like for collection techniques, it's very, very important collection technique. So we need to, if we can standardize the collection, is very good, but it's not easy today, because the medical technologies or nurse to take a swab is not very, very This is because this is new, so they just train for this. And type of specimens, because they can be different CT value. If we use saliva, nasopharynx, combined naso or oropharynx, they can different CT value. Of sample time, sampling time, of viral kinetic. For analytical factor, internal control, because some adjust by internal control, type of real-time PCR, purity of reagent, pipetting defect, and post-analytical about the interpretation of amplification to how to adjust base correction, error in transcription. This is a factor can affect CT value. And now just published about the viral load in infectivity, but this need depend, maybe some instrument different, so they can difference based on this publications. If the score is between 70 to 24, this high viral load. From 24 to 35 is moderate viral loads, and from above 36 is they say non-diagnostic result. But this is need to prove. We need to compare with other method, because we look at some method, they have different cutoff. Yes. So we need to compare. Maybe this is for 40 cutoff. And we need to know they have some technology, they have different sensitivity. Based on FDA data, they have a reference panels to share to another lab, and very, very huge difference sensitivity. Some laboratory can lower than 5,000 nucleic acid detection unit, like for Perkin, Hologic, Bioret on Rose, and some between 500 to 10,000, like for Abbott, Gene Expert, Kaizen, Sherlock, and some technology with higher sensitivity, and they only can detect above 300,000 NDU, like for Abbott Adeno, peptide nucleic acid, Lyra detect. So the method is very huge difference of the sensitivity. And meta-analysis, based on meta-analysis, sensitivity, specificity for, especially for POCT, they have different too, because some Indonesia laboratory, to small laboratory, they utilize or POCT system. They have two different types. Some POCT, they use purified RNA. Some purified, they utilize crude patient samples. So no extraction process in extract in external. They direct to crude patient samples. And for crude patient sample is the lower sensitivity comparable with the sensitivity in purified RNA. So this is very important to compare about that. And factor affecting CT value, this is I think double, sorry. And because to control, it's very important to control the results because we have a reference gene or housekeeping gene can be a control. Two types of control available: control as a bacteriophage as internal control, or RNAsP as an internal control. The limitation of bacteriophage internal control is because we cannot monitor the samplings. For RNA, we can monitor the sampling, but sometimes if the sampling is not so good, so we cannot monitor the process of extraction. This is a lot of type of control available. We can use exogenous control like bacteriophage, or we can use internal control. Mostly they have a specific primer, endogenous primer and probe. And after we extract, we have positive control, negative control, and sample. We are running same time with the samples, and we extract. To monitor this, we can add internal control with positive control, negative control, sample control with IC, and no template control of positive control. So we can monitor a false negative or false positive in our running molecular testing. And this is if we want to control a lot of all of steps from specimen collection, RNA extraction, amplification, instrument data operating reporting. This is a type of control to know the specimen properly taken. We can use IC human target. In the extraction process, we monitor with NTC to monitor contamination, or exogenous internal control, or endogenous internal control. For monitoring amplification, we can utilize positive control to monitor the false negative, or the real-time PCR work properly, and negative control to monitor the cross-contamination, and internal control to monitor PCR inhibitor. So each step we need to have a control, so we can make sure all steps we can monitor perfectly. And this is examples variation in the reagents. They have different internal controls. Like for a gene expert, they use exogenous controls, and for COBAS they use exogenous too, and for endogenous, like for CDC, use RNA as endogenous. This is a different LOD. And to monitoring false positive, we need to know the number of positive cell per matrix, number of cell cluster per matrix, so we can count how much this. If they have bigger, is potentially cross-reactivity or cross-contamination, because we need to count how much number of, how big the cluster. It's very important to monitoring false positive with positivity rate, especially in this Indonesian phase now, is because this is a very high case, positivity rate around 30 to 40% in the lab. We need to monitor this because mostly this is larger. So because 40% patient will be positive here, so the sometime cluster is bigger. So we need to define how much the cluster depend of the prevalence of positivity in our lab. And we need to make a KPI in our lab, like a number of specimen tests, you know, so it depends of dashboard. And we need to have to compare with the external control, especially for proficiency testing. Like in Indonesia, we collaborate with lead bankers of cytometric, SeraCare, or another Bioret, another control available for as a third party.
I'm almost finished.
Okay. The last slide is about the related to variations. I have 5 minutes, to closing this presentation. Variation can impact to the result. This is a different target of the reagents. And now we have tool to check the variations. So if you have a reagent, like for variation B117, this tools is checker tools to know what our reagents have impact to variant or not. We can check B117, check here, and we can check what reagent not impact. Like for this, for cephates, they have 2 genes, no S, 2 gene, no S with the reagent. For Perkin, they have 2 N's, but for Thermo, they have S gene. But if we look at here, they've already verified not affect to the variant. So we need to now based on these tools. Yeah, it's very important tools for all lab before they choose the reagent, or they want to know the reagent is now they have evidence related to variants. And to interpretations depend of the what we utilize. As a conclusion, variability in testing result has significant impact on patient care and manage COVID-19 pandemic. Main pitfall in SARS-CoV-2 testing are variability. Lack of standardization can increase variability interpretation. We need to harmonize all variable better way to health management pandemic. Thank you very much, Dr. Mark.
Well, thank you very much for Dr. Miswar. And now we are going to start with the questions. Maybe I think we start with Prof. Ferarri. The questions from the participant, please. Or we can play the recorded questions. Dr. Miswar, Prof. Prof. Ferarri.
Maybe from host to play the question one. Yeah.
Yeah. There are two questions from recorded. Two questions. What is the role of IACC in reducing variability of molecular test results? And the other questions, how to reduce variability with the non-standardized examination of SARS-CoV-2 RNA. And you can see the questions on below screen, please.
For sure, in this outbreak, IACC did a very strong active role because from the beginning, IACC set up a task, an important task related to the COVID-19 infection, and develop several guidelines for suggesting how to work in this field, publish a lot of papers with a lot of suggestions, and still in our website that is managed now by the Professor Adeli and the collaborators, there is a continuous update on information regarding the different approach, not only molecular test, but also information the serological test, vaccine, and so on and so forth. And this means that really IACC did a very, very important job to try to update every day, I would like to say, information for all laboratorians in all the world.
The next question, please.
This is an important task. I would like to comment immediately that because of the unbelievable growth of the infection and the clinical problems and the unbelievable short time to develop the test, we are very far for the standardization, particularly in the molecular test. What I mean that usually in our activity we need much more time. Now we have several hundreds of different kits. There is a different sensitivity. There is no is not fully standardized. This means that for sure the quality of the results are not standardized. There is a variability, but it was a fight against an unbelievable number of tests that we have to perform every day in all the world. What now we have to do is that because at least as much as the numbers will decrease, we have more time to move in the standardization, optimization of the test, and to move as the usual rules as indicated by IACC in one of the papers that we published.
Yeah, thank you for the answer from Prof. Ferarri. Some of the participants would like to ask the questions. And what about this, Dr. Miswar? we take notes and then we send it to him.
Yes.
Okay.
If other questions
so
from participant to Prof. Ferarri, we can send to Prof. Ferarri.
Okay. So I think I would like to there is a question from me about the kit. So the kit for the detection COVID-19 currently uses two target genes. Which of these genes combination is the specific or recommended? Gene E and RDRP, gene N and RDRP, and some use ORF1AB and RDRP. So and the question also, what is the principle of this combination? And Prof. Ferarri answer the questions. When you select primers, some rules should be followed. Very specific for SARS-CoV-2 virus to avoid Meyer pairing. Target very highly conserved region to avoid false negative. Then when you set up a kit or test, you try different combination and you apply the most robust and sensitive. This is the reason why starting very quickly, we have several combinations. Last moment, it's mandatory to target at least two regions to avoid false negative. And it's also important when we have a new variant to check is could be detected by our kit. I think some answer of this from Prof. Ferarri already mentioned by Dr. Miswar as well. Maybe you have any more answer, Dr. Miswar, for this?
I think enough for that question.
Okay.
Thank you.
Now, any other questions?
I think other question in chat, Dr. Damar.
Yes. Sorry, I can't read it. It's very small. Mungkin bisa dibantu, saya nggak bisa baca, ini kecil banget.
Okay, Dr. Demak, maybe I will read this question
Yes,
in the chat.
please.
This is from Mary Handayani. The question is how to minimize false positive in PCR testing.
I think you can answer,
yes,
Dr.
I can
Miswar.
answer. Yeah, thank you. Thank you, Dr. This is very important because especially in the very, very high case, the potentially positivity, false positive will be increased. The pipetting error, how to utilize pipetting is very, very important, very source, and how to manage the samples if the sample is very, very close contact with others. It's very easy to become a cross reactivity. So this potential for false positivity. And include how to manage the position of positive control, or we manage the positive control, because this positive control, they have very high number of DNA or RNA in that control, so they can become cross contaminations to other samples. And we need to make monitoring the potentially positivity rates. So we need monitoring daily by day or batch between batch, how much the positivity rates, include the positions in the wells. If they have a high cluster, it's potentially, if they have patterns with the very, very close in the cluster, it's very potentially false positive and because the cross contamination. Thank you, Dr. Mark.
Yeah.
You can read another question.
No, please read for me.
Yes. Another question is from Nurmalia Purnamasari, how to use positivity rate for monitoring false positives. Very important about this, we need to know what patients we have, because some patient only for control, some patient for diagnosis. So the positivity rate between laboratory depend of the patient. So we need to monitor batch between batch and daily by that the positivity rate. We can compare with other lab, because now very easy to get the positivity rate other lab, and include the national positivity rates. We need to compare and we need to monitoring the trends. If the number case is lower and potential or patient is not very high, the possibility of positivity and our case is high, so we can must make additional control to monitor the potentially the false positive. It's very important to monitor false positive in our laboratory. And another question is from Afinas Ramasarap Hadun. Sorry, maybe I not easy to read the name. How the ideal time for nasal swab and post-infection, how much time for real-time PCR to come negative. I think this is similar with Prof. Ferarri today already informed too about the ideal time for nasal swab. Ideal time is if we have a good sensitivity or instruments, we can use a nasal swab. And the type of nasal swab is if the higher peak is very important, but we don't know the higher peak of the patient. If they have symptoms, it's very good to it's very best time to take a nasal swab. For instance, how much time for real-time PCR based on average, the between 1 month to 4 weeks, the positivity rate will become negative. This is average, but some patients they have longer, depend of the biological of the patients. But some issue about this is potential only the fragment, not actually full virus about that. Thank you, Dr. Demak. I think there's question input in the chat, so no other question.
Any other?
No more, Dr. Demak, in this chat room.
Do we waiting for another questions? We still have time. Well, if we don't have any more questions from the participant, so we end this session. So finally, we can finish the session. And thank you very much for to both speakers for their interesting and useful presentations. And I will read a little summary of these sessions. Prof. Ferarri already give information about the variety of molecular testing and interpretations, and also it is about the guideline from the IACC, and is also mentioned about the critical role of the lab in COVID, and also he mentions several genes targets recommended by the WHO, and also we have to pay attention on potential pre-analytic and analytical variabilities, and also he mentions several reverse transcript PCR and also how to standardize them. And he mentioned also about several PCR, isothermal, non-isothermal, and also mentioned the sequencing, kemudian, and then prolonged significant of the positivity, and also a little about COVID in variant and how to detect the variant. And from Dr. Miswar, it's about the variety of, pitfall of the COVID-19 molecular testing and interpretations. And a moment, please. Yeah, he mentioned about the epidemiology of COVID-19 in Indonesia, and also mentioned about now quite a lot of lab for PCR from 32 to 33, and now most of them only have one instrument, and it is also the pitfall of the working of the PCR detection. And then he also mentioned about the collections of samples that we have to pay attention for the correct test. And then you also mentioned about the critical factor. I think this is the most important, about the degradation of RNA, and then a PCR inhibitor, and low quality of reagent, low concentration of RNA, and then pipetting error, it's often, and contamination, and quality of the PCR tube and well, and also low quality of a high light cycler available. I think it is the very important. And other Dr. Miswar also mentioned about the ideal design for molecular lab. That's all the presentation from the both speakers, and I think I would like to say thank you.